All right, I am recording this because my plan is to post this kind of overview video of the lab and also of the first lab, because the Thursday lab does not meet because of the academic holiday. And apparently my cat is going to try and be in this lab the whole morning with me. So this is Ginger. She is very needy when I'm at home, which is why I often like to go to my office, but I do have virtual labs today. So today is my day at home. Okay. All right, so let me get my screen share going. All right, here we go. Okay, so this is a PDF of our course syllabus. This is available for download via Canvas. So you don't have to take notes on this, but I kind of want to go over it briefly so you have a good understanding of what you'll be doing this quarter, specifically in the lab piece. So your lab has several objectives. So you're going to be working collaboratively, and this is going to help you, build your skills in learning how to work collaboratively with a team of scientists. You're going to gain an understanding of the state of research in a particular area. As I mentioned in the lecture, you're going to pick a research question and you're going to work with your group all quarter on that research question. You're also going to design a research study to address a particular knowledge gap, which we'll talk about more. You're also going to conduct basic statistical analyses. So we are going to be using a statistical software called JMP or JUMP. Did you guys use that in 298 at all? So hopefully I see a little no. Okay, no. Okay, so it's okay if you haven't used it before. We're gonna learn how to use it this quarter. So if you don't know how to use it, that's totally fine. I do have instructions on Canvas for downloading JUMP. So you will need to download that. Everyone should download that to their computer this quarter, preferably sooner rather than later, because you're going to come up to a lab where you're going to have to use JUMP. And so you'll need it on your computer before that lab. And then the last objective is to present findings of a research study. So again, you're going to be kind of working on a research question all quarter, and you're going to analyze data that you collect, and you're actually going to present the findings from your analysis in writing and also in an oral presentation format. So we've got 10 weeks of lab. This first week, we're just doing some practice problems, but you'll see in the other weeks, you've only got practice problems. I think in week 5 is the only other week that you've just got some practice problems, but the rest of the weeks, you'll be doing work that is related to your group project. So we'll go through those each week, and I'll try and release the instructions in advance on Canvas so you can read through those before lab. And these labs are worth various points, as you can see here on this column. Okay, so you also have a team project that you're going to be working on, as I mentioned earlier, and we also briefly talked about this in the lecture. You should, with your group, have your research question identified by week 2. So I highly recommend identifying your research question before you come to lab. I think I mentioned in our lecture that there have been, instances where students have come to the week 2 lab to start talking about their research question, and then they spend a significant amount of time in the week 2 lab talking about what they're going to do, and then they kind of run out of time for the actual lab in week 2. So I highly recommend thinking about your research question in advance. You're welcome to email me if your group wants to run it by me in advance. I'm happy to talk with you about your research question. And what that means in terms of developing that research question is you're going to need to identify an independent variable and a dependent variable. So your independent variable can really be anything you want. There's some examples here. So physical activity, sedentary behavior, diet, smoking, drinking, vaping, sleep quantity and quality. A lot of groups are always every quarter really interested in sleep. And so sleep is a great variable. Social support, perceived stress, self-reported health, employment status, all of those would be great examples of appropriate independent variables. And you can also pick some of these for your dependent variables as well, so your outcome variable. The only caveat with the outcome variable is that it has to be binary. And so that doesn't mean you can't pick something like sleep quantity, so maybe hours of sleep. That is a continuous variable by itself, but you can make that variable dichotomous, right? So you can draw a threshold and say, anyone with 7 hours or more has good quantity sleep, and anyone who's sleeping under 7 hours has maybe low quantity sleep. So you can always make a continuous variable into a binary variable for your outcome. So think about what variables you're interested in assessing and seeing. You're essentially examining the association between those two variables. Another one students often do is GPA. So you can look at GPA as a variable in your research question. It really is up to you, but you want to be cognizant of the variable you're choosing in relationship to your sample. So what I mean by that is I've had students in previous quarters want to assess the association between gender and physical activity. One of the challenges that we often face in this class is that you are surveying the students in your class and maybe hopefully some people outside of the class as well to increase your sample size. But when you look around in the class, there really aren't a lot of people who identify as male. The class tends to be predominantly female. And so if you have basically no males in the class, it's impossible to assess gender as a variable in your research question. So you want to think about a variable where there's going to be some kind of variation and distribution in that variable. So that you need to have that kind of solidified by your week 2 lab, because in week 2, you're going to be actually doing kind of a literature review and finding articles to help write the introduction piece of your project. So again, I'm happy to chat with you. And if you want to email me and throw an idea by me, you're welcome to do that. And I can help you kind of finalize your research question. There's also instructions in here on the final report that you'll need to complete with your group. Your final report is going to be written like a manuscript that you would find on PubMed. So you'll notice that there's an introduction, there's a methods section, a results section, a discussion section, and there's also some other things that are required in here as well. But essentially, it's the format of a published manuscript that you would pull from PubMed or another database. And then you also, in addition to that written report, you have an oral presentation. It's pretty short that you'll give at the end of the quarter that you'll present to the class on your project and what you found. Okay. Any questions on the lab and expectations around the lab? All right, let me see. Yeah, and so someone has noted that the JMP download instructions are in week 2. I'm going to be unlocking week 2 very soon here, so you'll have access to those pretty soon. In terms of your weekly lab assignments, they're due after lab. So you should use the lab to actually work on your weekly lab, and they're due, I think I have them due like 15 minutes after our lab officially ends. So our lab ends at 11, so that would be due at 11:15. What I will say is that there are certain weeks where it sometimes takes some groups a little bit longer to get through the lab. That's okay. If your group is here and you're all working together and you just run out of time and you need some more time, let me know. I'm happy to extend the deadline for your lab, but I do need your group to let me know that that's something that you need, and I can make that accommodation happen. I don't want any situations where you're getting late points taken off for your lab. So again, I'm happy to work with you if your group does need some additional time to complete that lab. That doesn't happen a whole lot, but every once in a while it does happen. So just please keep kind of open communication and let me know if that's something that does need to happen. Okay. So that is the kind of the overview of the course and the syllabus. I am going to share our lab 1 and we'll briefly go over that. Okay. So this is your first lab. So the lab you're going to do this week. For all of your labs, you're going to be turning in group submissions, but I decided for this first week, you're going to turn in your lab individually, specifically because there's not a Thursday. Thursday's a holiday. So the Thursday lab, I'm not sure if everyone's going to get together with their groups. I'm hoping they do. But because there isn't an official meeting, I've made this an individual assignment, but I'm hoping that the Thursday lab, I hope everyone still works together. They just find a time to do it on their own. But this lab in particular, you're going to submit individually, but you should work with your group to complete it. Okay, so this first lab are just some practice problems to kind of refresh your memory from last quarter. So none of this should be new information. It's kind of just a review on some things that you've already learned. There is this lab document, and then there's also an Excel document that you'll download. Some of your answers you're going to just type in using red font into the document. Otherwise, if it says to use the Excel sheet, you should use the Excel sheet and put your answers in the Excel sheet. So you'll have some answers in the Word document and answers in the Excel sheet, and you'll submit both of those with your lab. Okay, so in this lab, we're going over a lot of things that you've kind of already learned, right? So things like incidence and prevalence, hopefully those are terms that sound familiar to you, but there's some instructions here that you should read through. And this is kind of a refresher on what these terms are, right? So prevalence is essentially, it refers to the number of cases in a population at a given point in time, right? So it's kind of the proportion of that population that has whatever the disease is or the outcome of interest at a given point in time, right? And incidence is different, right? That's the number of new cases during a certain point in time. And these numbers mean different things, right? And they're calculated in different ways. So with incidence, we're looking at the number of new cases in a population divided by the number of people who are at risk of getting that disease during that period, right? Versus prevalence, which is essentially the proportion of people with the disease, the number of cases in that population during that period of time, right? So the denominators of these calculations are slightly different, right? And the numerators are also slightly different. So your prevalence is kind of just the proportion of that population that has the disease at a given time, while the incidence is looking at the number of new cases divided by the people who are actually at risk of getting that disease during that period of time. So that denominator in the incidence calculations, as you go through these practice problems, that denominator might look slightly different for the incidence calculations versus the prevalence calculations. So you want to be cognizant of your denominators that you're using and also the numerators in these calculations. So we often like to adjust our calculations per X number of people. So we're able to kind of compare those across different groups. And so this is just a reminder on how you can express things per, for example, 1,000 people. So you're going to need to do that in your lab. So these are just some example, the formulas that you can use to actually calculate those numbers. So for example, cumulative incidence per 1,000 is going to be that incidence calculation times 1,000, and same with the prevalence. Okay, so this first kind of example here, I think this table is also in your Excel document, so you can take a look at this. This is just showing you how they got all of the different numbers in this table as the years kind of progress, right? So this is at year 0, this is at baseline, this is year 1, 2, 3, 4, 5, and you can see that you're getting all these new cases as the years go on, right? So in year 1, there were 55 new cases, year 2, there were 50 cases, and so on. This column is just telling you the total number of cases, right? So it's just adding in the cases that have happened as the years go on. These are the individuals still at risk, and this is showing you that annual incidence calculation, right? This is the incidence per 1,000. So they just use that formula that we just reviewed. And then this is the prevalence at that given point in time. And then this is the prevalence per 1,000. So this table is just showing you how they got all of these calculations to kind of refresh your memory on how to actually do this with a specific example. Okay. So you're going to kind of do a lot of these calculations through various practice problems. There's 5 problems in here that you're going to work on. The first one is a practice problem around autism. So you'll read through this and they give you some data here that you can work with to answer the questions. There's a spring fever question where you've been given some data and you're going to need to calculate the numbers in here. This table should go into your Excel documents and you'll fill in these numbers, right? So for part A, you're going to calculate the prevalence and incidence rates and express them as decimals. So you'll do that and you'll fill out this table. In part B, this is just asking you to then in your Excel document, add columns next to these. So you'll add a column here and then a column on the other side that then adds that incidence per 100 and the prevalence per 100. So you should just add that to your existing table so it's all in one table in your Excel document. And then you'll answer part C based on the numbers that you calculate. And then in question 3, you've got a question on diphtheria. You'll use the Excel document to answer this. You'll go through here and you'll answer these questions and follow the instructions in here. For part B, you do need to create a little graph in Excel. And so when you get to that point, if you need some help with that, let me know. I'm happy to help you. And then in part 2, you're getting into measures of association. And so in part 2, this is kind of just a practice example that walks you through an example of how to calculate those numbers and what exactly that means. Hopefully you all remember 2x2 tables. If you don't, this is going to refresh your memory on 2x2 tables and how to do those calculations. And so this is just part of the example in here. And you'll read through that. This is, I think, another example problem that they give you as well. So there's lots of examples to go off of in here. And then you'll get to this worksheet 2, and now you'll actually have the practice problems that you're going to work on. And so this first one is looking at the risk factor for better grades. So you'll need to set up a 2x2 table in your Excel sheets and calculate those numbers based on the information you're given. And then you're going to look at an exam or a question on smoking and duodenal ulcers. And so you're going to follow the instructions in here. You're going to answer the questions here. You're going to need to set up 2x2 tables and do those calculations for that as well. So that's kind of the plan for our first lab. Are there any questions?
I had a quick question.
Yeah.
Do you think you could sort of reiterate the difference between the denominator and prevalence and incidence?
Yes, so let's go to that table. Let's see what that table is, that example. Oops, I think I passed it. Hold on. All right, this one. Okay, so this table shows you a bunch of different things, right? But it's got essentially each year, and it's got the data from each year, right? And so this is telling you the number of new cases in that year. So in year one, there were 55 new cases of whatever the disease is. In year two, there were 50 new cases. Year three, there were 55. 4, there were 50. And in year 5, there were 55. This is telling you the total cases so far. And this is telling you essentially at the end of that period, how many people are still at risk. Now, what you'll notice here is that this denominator in the annual incidence, this is changing, right? The denominator in the prevalence, is that changing? It's not changing, right? So that's one of the big differences in those calculations, is that the denominator for the incidence is going to change because you're looking at the number of new cases divided by the people who were at risk of developing the disease, right? Versus prevalence, which is essentially just saying it's the proportion of people in the population that have the disease at a given point in time. Does that make sense?
Yeah, thanks.